So I'm Taylor. It's the last session of the last day of the conference. I really, really appreciate you showing up for this. I'm excited to talk to you about some research that I've done with Martin Turcotte. I'm going to try advancing this. Mart is a proud Montreal native and francophone himself, and therefore mandated, required that in keeping with local law and custom that I display this. Now, I am not a French speaker myself, but I think what it says is that I'm excited to talk to you about an experiment that I did trying to investigate how experimental communities of plants evolve in response to novel invaders. Let's see if it advances. And I don't think this is novel information for this crowd, that invasive populations evolve in response to novel threats and interactions in their introduced ranges. You know, invaders respond to different human interactions, their environments, and the residents. These can all pose strong selective threats to invaders. But invaders, at least successful ones, are kind of notorious for punching back. And when they do this, they can impact humans, they can impact their environments, and they can also impact the residents themselves, causing a potential being a potential selective agent. And it's this part of the relationship or interaction that I want to focus on. Because if there's an evolutionary interaction here, this could have long-term consequences for how those resident communities ecologically respond to the same invaders or other invaders in the future, potentially shaping the community's invasibility. And thus providing kind of this indirect long-term eco-evolutionary consequence that could be really difficult to measure and assess in nature, but experimentally kind of cool. So really what I'm interested in is kind of like completing the circle. So yes, ecology can dictate evolutionary outcomes, shape differences in things like traits, alleles, compositions, and frequencies. But does it have an impact? Does it change the ecology of these communities? That's the part that I'm really interested in. And in particularly for this experiment, I'm interested in how that might shape the community's invasibility. So invasibility is kind of a funny word. It's not something we use on an everyday basis. So by invasibility, I just mean the differential susceptibility to invasion by outsiders. And you could think that maybe two different communities invaded by the same species might have some differences. We can measure these differences, and that's kind of, I'm teasing a little bit about the kind of data I'm showing you in a few slides. That's what we're measuring. And in particular, we're measuring how evolution influences this. So the questions that I set out to address, or at least what I'm going to be presenting here, are really interested in understanding, do invaders cause differential resident evolution? And if so, does this evolution dictate changes in the community invasibility? So to do this, I was using a model system called duckweed. Duckweed is a small floating aquatic plant native to North America. It is very tiny. It is very fast growing, so generation times of as short as 3 to 4 days. High degree of heritable genetic variation, which makes it a really good system for studying evolution. And we can do these large-scale experiments with hundreds or thousands of individuals, so large populations, large communities, and we can do them in semi-natural environments because we want to invite with that noise and that realism into our experiments, because noise and realism is what happens in nature. Now, for our invaders, we used a couple species of invasive Salvinia. Salvinia is a floating aquatic fern. Quick shout out to Nick, actually, who I attended CPING a couple years ago. It was Brittany Sutherland who introduced me to these, so that was really cool. Thank you. These are native to South America, invasive in the American South. They have a very similar natural or life history to duckweed, so they're free-floating, grow clonally, vegetatively, and they're really problematic. They're known for outcompeting native vegetation, native aquatic plants in the South, but notably, at least in the watersheds and water bodies where we collected our duckweed in western Pennsylvania, there's no overlapping or no co-occurring evolutionary history. So these are very novel invaders and very novel competitors for our genotypes. Okay, but in order for evolution to occur, or if we want to predict evolution to occur, we suspect that there should be some genetic variation in this, I don't even want to call it a trait, but this property of invasibility. So to help find out if that was possible, I conducted an experiment with undergraduate Ashlyn Moretti, where we looked at how different genotypes growing alone with the invader, Salvinia molesta, how they were shaped into differences in invasibility. And what we see that there's like a high degree of difference in how susceptible they are to invasion. Thus kind of informing that maybe this is like a heritable characteristic, something that could be passed on and possibly under selection. So in this experiment, what we did is we started out by evolving our populations and communities of our native species in the presence or absence of Salvinia molesta, Salvinia minima, a combination, or neither. Now, we did this for 8 to 12 generations. What did that look like in practice? So we used 7 genotypes of each of our native species at equal frequencies in these 1-gallon buckets, so pretty large for, relative to the size of these populations. And then in some cases, we invaded them with Salvinia at about 1/10 the biomass. We let them grow for 7 weeks at the Pymatuni Lab of Ecology in northwestern Pennsylvania in mesocosm conditions, cattle tanks, and monitored them. Now, we could stop there and just look for any differences in the ecology and evolution of these communities. But because we wanted to understand whether any trait changes or properties are the result of genetic change, we needed to do a common garden. And to do this, we separated everything we put together, we had to pull out. And so we separated our populations of our natives, and I can confidently, wholeheartedly say this was, without a doubt, the worst part of my PhD. It's like separating grains of sand from tinier grains of sand. I think Sisyphus would have been like, dude, what are you doing with your life? It was just brutal, but with the lows come some highs. And this experiment really afforded me the opportunity to work with just a phenomenal team of undergraduate research assistants who helped pull this kind of miserable thing off and make it not miserable at all, and actually really fun and rewarding. And so I wouldn't be standing here without the data that they helped me collect, and also emotionally without their support. So thank you to them. So to answer this question, do we see any signatures of evolutionary change? So following the common garden phase of this experiment, we phenotyped all of our populations. And so what I'm going to show you is just kind of this composite for size, so biomass and surface area, looking for any evidence that these populations differed from each other. So for Lemna minor, our smallest duckweed that we use in this experiment, remember these are the natives, we see that relative to our non-evolved, a composite of the non-evolved genotypes that we use, everything evolves to get a little bit smaller, but it's only those populations that evolved with Salvinia minima that get significantly smaller than those that hadn't evolved, or the ones that evolved with Salvinia molesta. So it appears that there's this important thing going on. We're seeing evidence of possible genetic change. Their phenotypes are different. Now, for our other native species, Spirodella polyrhiza, the pattern's opposite. Everything gets a little bit bigger, but it's only for the surface area, and it's only those that evolved with both species that get significantly larger. So evidence of phenotypic change. We're analyzing the genetic results right now and kind of excited to try and corroborate those to see consistencies or inconsistencies. But so what? They evolve, their phenotypes are different, but does that shape how they interact with the invaders themselves in the future? So to do this, we reconstituted those same communities from before, from our common garden populations, into new communities where they were invaded by new Salvinia, where they experienced both the same invader and different invader combinations or single invaders that they had before. This allowed us to test for kind of evidence of local adaptation to one invader versus kind of a general overall invasibility change. And we did this for all of our treatments. It was something like 300 individual communities that we measured. These were established and grew over the period of 2 weeks, and after setting them up, we deconstructed them again and took change in invader growth over time. And so that's what I'm about to show you now is how they differed in their invasibility by measuring this proxy invader growth. And I'm gonna kind of spoil the result a little bit and say that we did not see any evidence of local adaptation. It did not appear that evolving to one invader made them better at that invader and that invader only. But what we did see is we did see the signature of potential change in overall invasibility, such that those communities, and I remember not just populations, but communities that evolved with Salvinia minima were significantly less susceptible, less invasible than those that evolved with Salvinia molesta. And so it appears that they're like compared to those that evolved without Salvinia, they're kind of going along these separate trajectories. There might be this evidence of possible evolutionary priority effects occurring. And remember, these are being invaded by members, like, yes, they're morphologically very different, but they're also members of the same genre. So, you know, they're both part of Salvinia, so, but very different outcomes. And there's also a tremendous amount of variability here. I kind of expected this, but I just want to point out that, like, not every community is going along the same doesn't exhibit the same community properties. And if you think about it, these are complex communities with so many different individual, like, pairwise interactions between genotypes of the same species, the same different native species, and the invaders, that I kind of expect this noise to be here. But I'm excited to get some of that genetic data back to try to understand if that helps explain some of the variants that we observe. So we do see some evidence that there's this change in overall invasibility. And some final parting thoughts. You know, this could have implications, you know, if we're thinking about evolution. You know, what are these consequences of evolutionary priority effects? You know, thinking long term, you know, they experience one thing, but generations later, there can be long-term consequences. And for biological invasions, I had never really thought before that invaders across a landscape might be serving as sources of evolutionary diversification of our native communities and populations. This could be, like, really, really hard to measure and track, but it could be happening. And I don't think it's a good thing, but I think it's something to be thoughtful of. And for conservation, you know, if we still don't know mechanistically how our communities became less invasible, but maybe it's because they're becoming more like the invaders. Maybe they're competing more directly. And if so, are they evolving to become more invasive themselves? Does that shape the way in which native populations interact with each other? So it's kind of like, you know, there's a lot to unpack here, and I certainly don't have all the answers, but I think there's also a lot of avenues of further research. A ton of people have helped me throughout the years. And right now, I just want to say, again, thank you so much for being here. And I'd appreciate any questions you have. And if don't get them in here, come talk to me at the social. I just love getting to meet new people that do cool stuff. So come chat.
Yeah, I was wondering if you could reflect a little bit on sort of the role of the microbiome here. Do you think, is it like the actual plants that are evolving, or is it the microbiome that's changing?
So the question was about how the microbiome might be influencing, and I could imagine that could be through ecological or evolutionary change of the microbiome as well. In this case, we did everything possible to eliminate that as a factor. We used we bleached and sterilized all of our lineages. Now, is it possible that some of that escaped, or they acquired new microbiomes at the time? You know, at the time, we tried to treat everything very similarly to prevent that. But we do not have any data or evidence, and nothing visually striking, but I'm not sure, as I'm not a microbiologist. We didn't see anything that would make me suspect it was, but it's possible.
Hi, everyone. Thanks for being here. I really appreciate you guys coming out to see us. Okay, so today I'm going to talk about some of the work I've done with Mason Heberling. He's at the Carnegie Museum of Natural History, and Rachel Reeb at the University of Pittsburgh. And we worked together on a project that we're now in the process of writing up. Okay, so first, let's get into the weeds. So this is a photo, thanks. I do that joke every time, and it always kills, guys. this is a photo of an agricultural field that's infested with blue mustard. So that's everything in this photo that's purple. And when I see a landscape like this that's really dominated by a species we didn't intend to put there, I want to know what the heck is going on. And that leads me to my motivating question, which is how do invasive plants succeed in novel environments? So by invasive species, just to be clear, I mean a non-native species which adversely affects the habitats it invades. So they're highly problematic, but they're also these great evolutionary test cases, I think you guys will agree. And I think of invasion biology as the biology of winners, and I want to know how they did it, right? So with invasions, we can ask questions about all of our favorite evolutionary processes, how they might impact these plants in novel environments, and some of the questions that we want to understand are invasive species invasive before introduction? Or in other words, are they pre-adapted or exapted to similar environments, and that's what allows them to hit the ground running in these new habitats? Or do introduced populations evolve in these novel environments? Do they evolve to be more invasive? So one way we can try to answer this question for a particular species is by collecting individuals of that species from its native range, collecting individuals of that species from its invasive range, and comparing the two, and saying that any differences we see are due to evolution in the invaded range. And I've done that myself in previous work. But I want to know if we can make better comparisons, because there are some challenges with comparing contemporary individuals from the two ranges and just assuming all the differences are due to evolution in the invaded range. So it's often not clear what exactly we should be comparing those invasive populations to, like which are the correct native populations for that comparison. This can be complicated if multiple introductions are involved, which seems to happen a lot, or if we lack detailed population genetic data, which is true for most invasive species at this point. And additionally, it's not like evolution has just stopped in the native range, right? The Anthropocene is happening everywhere. Novel environmental conditions are occurring, basically everywhere. So ideally, we could observe the direct predecessors of the contemporary invasive populations that we're interested in. And what we need for that is a time machine, essentially. And we kind of have that conveniently in the form of herbarium specimens or other natural history specimens. So natural history collections are really rich sources of the primary data collected by the original collector. So that information you'll find on a specimen label, which on its own can be super useful. But what I think is really exciting, and that more and more people are starting to do, is that you can get even more data from these specimens. So this new data can be collected from the physical specimen itself, or from digital representations of the specimens, and that makes them very accessible. And one of the many types of data that you can get from a digital image of an herbarium specimen is trait data. And in particular for this project, we were looking at phenological trait data. Okay, so please take a moment to appreciate my joke title. If I don't point it out, then you wouldn't see it. Okay, folks have really become interested in the last, couple of decades in investigating phenological shifts. In other words, the timing of events within an organism's life cycle and how that has changed over time, particularly in response to climate change. Herbaria and other historical data sources are really important for these kinds of investigations, which have often shown evidence of rapid evolution in phenology in species in response to climate change. Somewhat less work has been done on the phenological differences that might exist between the native and invaded ranges of a species. And I can actually only think of one study that's looked at phenological shifts occurring within the invaded range of a species. But if you guys know of any more, please tell me. Okay. So that's our setup. Let's meet the star of our story. This is Chorospora tenella, or blue mustard. A few things about this species make it really convenient to work with. First of all, it's weedy. It's a weed of winter annual cereal crops like winter wheat and alfalfa. And it's also a weed of arid rangelands and roadsides. It's one of about 3,000 species that have been designated officially as a weed by the Weed Science Society of America. And to date, most of the work that's been done on this species have really just been looking at what herbicides can we use to kill it, like how much does it take. And also the species has been identified as having traits that are highly associated with herbicide resistance, suggesting that while we don't have evidence of herbicide resistance yet in the species, it could occur any minute now. So it could be a growing problem. Okay, it's invasive in North America. It's native originally to Eastern Europe, but also the Balkan Peninsula, Turkey, Iran, Mongolia, northwestern China, and northern India, so a pretty wide range there. And it's been introduced to North America. It's currently observed in at least 32 states and 3 Canadian provinces, including everywhere west of the Mississippi. And it's particularly concentrated in the western arid counties of the US. It's also well represented in North American herbaria. So it was first introduced at a point when there were botanists actively collecting, so we could collect it throughout the course of its invasion. It first showed up in the very late 1800s, as far as we know. And indeed, botanists did collect it. So this is the cumulative count of herbarium specimens listed in at least this one large aggregator database, which lists about 1,700 specimens. Okay, so to return to my motivating question, I want to know how this invasive plant managed to succeed in these novel environments. And one of the things that you can do with herbarium specimens is to ask questions about the abiotic environment that these plants experienced. Each herbarium specimen is kind of a snapshot of a time and a place where that plant occurred. We can use just even that information off a specimen label to address questions about how individuals are distributed across a landscape or through abiotic environments, which is particularly powerful when combined with large environmental data sets. But we also have that individual right there on the specimen, so we can get trait data for those individuals. You know, did it have flowers? Did it have fruits? What time of year was it collected? And these are all questions about its phenology. Okay, so we've been trying to see what we can learn from the data preserved in the herbarium record for this species. And this is some of the raw data that we collected from some herbarium specimens. With this type of data and some really industrious undergrads, we can calculate a phenological index, so a quantitative metric of where each individual was in its life cycle when it was collected. And with that index, we can ask, does reproductive phenology change over the century-long course of invasion in this species? Is the species adapting to the novel environment by shifting its phenology? Because that would be pretty cool. The data set for this project includes 528 images of herbarium specimens from multiple herbaria, from specimens collected in the western US between 1920 and 2020. And we use the CREW monthly climate data as our large environmental data set. So here you can see our collections colored, or our specimens colored by collection year. I'll just cut right to the chase and say that we didn't actually find a pattern of phenological shift over the course of invasion, possibly because we would need probably need much denser sampling, especially in the early years, to establish that kind of pattern. But we did have enough specimens to look at how does regional climate influence phenology in the species. So the 2 climate variables that jumped out of our analyses as the most predictive of phenology were mean annual minimum temperature and interannual temperature variability. So mean annual minimum temperature was calculated across the entire climate record that we used. And then interannual temperature variability is the standard deviation of that mean annual minimum temperature. So the response we found was actually an interaction between those 2 climate variables. What we found was that the average timing of phenology across all life stages occurs earlier in warm regions than in cool regions, which is maybe not a huge surprise. But this interaction with the variability of that temperature variable is interesting to us. So this is a plot of the model outputs with mean annual minimum temperature on the x-axis, and the phenology day of year on the y. So the 3 lines represent different amounts of interannual temperature variability. Plants in the coldest and most temperature variable regions of the range, so that very top part of the green line there, those were the ones that experienced the most delayed phenology. So we speculate that this serves as a strategy to avoid winter frost. On this map, the plants in the locations with the yellowest dots reach a given phenological stage about 100 days earlier than the plants in locations with the reddest dots. So interannual temperature variability was less deterministic of phenology in the warm parts of the range, the warmer parts of the range. Plants in warm regions may never actually experience frost, so whether or not it's predictable might not have a huge impact on its phenological timing. And the phenological variation along this mean annual temperature gradient is contingent on the predictability of the temperature regime itself. So as far as we know, that kind of pattern hasn't really been shown in this type of system before, and it may have important implications for this plant under climate change, which should both increase temperatures, but also increase unpredictability across this range. We think that this spatial pattern might be driven in part by adaptive genetic variation in the phenological strategies that the species uses. And potentially, we can eventually identify the underlying genetic variation there, since I'm also actively trying to get genomic data from these very same herbarium specimens, which I don't have that data yet, but it's going to be cool when I get it. Okay, so I'm just going to conclude here with one of my favorite herbarium specimens collected by someone very fancy. I thought it was hilarious. Okay. Thank you. Okay, so to wrap up, I want to highlight that I'm on the lookout for more studies looking at phenological shifts over the course of invasion. If you know any, you tell me. That'd be great. And in Chorospora, we didn't find a phenological shift over the course of invasion, but instead a shift over environmental space. So to us, this suggests that while invaders might need to be plastic in order to move into these new habitats, they perhaps can also rapidly adapt to the unpredictability of the climate regimes that they experience there. Okay, and thank you. Oh, wait. I'd like to thank my collaborators, the folks that work in my lab on Chorospora, the herbaria that I've worked with, which have all been very generous, and also my funders. And I'd like to acknowledge the Shoshone and Bannock peoples on whose traditional land I now do my work. And thank you guys for listening. Yeah.
Is there a lot of variation in the current phenology of flowering time in the populations we see out there?
Oh yeah, absolutely. So there was stuff, so I'm in Idaho, and while it was still snowing in Idaho, it had started to flower, and in Nevada at the same time this year, it hadn't started flowering yet.
Like within a population?
Oh, within a population. You know what, I haven't surveyed that, I'm not sure. Yeah. Okay, thanks.
All right, hi everyone. The chime system is not working, so we're just on the honor system, I guess. I'm Azwad. I'm a PhD candidate at Cornell in Nina Turkelson's lab, and I'm going to shift gears a little bit. We're going to move on to fish, do some pop gen with them. So it's pretty clear that humans affect species and ecosystems in a variety of ways, whether it's through direct exploitation, the spread of disease or disease vectors, or anthropogenic shifts in the environment. This is the Invasion Biology Symposium, and it's really clear that humans are a major driver of evolution across many species and systems. And I'm particularly interested in biological invasions as a way to study rapid evolution, and hopefully you are too. And this is because invasive species can be thought of as these really amazing natural experiments in rapid evolution. This is because they allow us to study evolution in real time by exposing species to new environments and selective pressures. So in this cartoon example, we have interactions between the invader and the invaded environment and native species. This in turn can impart novel selective pressures on the invader and can result in altered phenotypes and traits and potentially local adaptation of the invader to this new environment. And what's really cool is that these transitions can occur really quickly, often within measurable or historically documented timescales. And that, of course, leads us to ask what sort of evolutionary genetic factors may underlie invasive success. So enter the American shad. At this point, you may have seen a couple other talks about this fish. But if you haven't, it's a member of the herring family. It's a clupeid, and it's anadromous, meaning it migrates from fresh water to the ocean and then back to fresh water in order to spawn. And individuals typically return to their spawning sites with high fidelity. So despite having a very large Atlantic range, you can still genetically trace them back to their rivers of origin because they tend to breed within the same rivers. So I'm interested in their introduced populations. So they were brought over in the late 1800s first to the Sacramento River and then later to the Columbia River. And they established in these rivers fairly well, but then they also spread very quickly across the entire West Coast. So they were found as far south as Baja, Mexico, and as far north as Alaska within a couple decades of their introduction. And now they have annual runs in San Francisco Bay and then further north in the Pacific Northwest. So there's like these contrasting fates of the populations in the native range and then in the introduced range. So in the native range, there's been a fisheries collapse around the turn of the 20th century. But in the native range, they're actually outnumbering all native salmon going through the Columbia River. There's literally millions of these fish, and they're doing quite well. And to tie it back to our previous question, what are the genetics underlying this invasive success? So with that, I have three sort of major framing questions. So the first is, do we see evidence of a founder effect? Typically, with invasive species, you're subsampling a much larger population, and that can result in reduced genetic diversity. And on top of that, can we actually trace the demographic histories of these invasive populations since their introduction? Second, what are the patterns of genetic structure among these invasive populations? How genetically distinct are they? With these populations, you have to wonder whether there's been enough time for drift to actually act on these populations in order to have a discernible signal. And then finally, do we see evidence of local adaptation? Are there these apparent islands of divergence between ancestral and introduced populations that may be indicative of adaptation to these new environments? So in order to address these questions, we sampled 340 individuals across 11 different locations on both coasts. We sampled 30 individuals per river and generated low coverage whole genome sequencing for all of these individuals, which resulted in a little bit less than 5 million SNPs after a bunch of filtering. So first we can check if there is any evidence of a founder effect. So we typically expect introduced or invasive population to experience declines in genetic diversity due to these population bottlenecks of subsampling larger populations. However, in some cases, you actually do find that invasive populations have even more diversity due to admixture or serial introductions. In this case, however, we have a pretty clear signal of reduced genetic diversity in these specific populations. So they have reduced heterozygosity compared to the Atlantic, and when you look at Waterson's theta, it's also significantly reduced. When you calculate Tajuma's D across the entire genome, we see that Pacific populations have positive value, which indicates a scarcity of rare alleles and can indicate that a recent population contraction. So overall, these results are consistent with a reduction in diversity, but does this, can we actually date the bottleneck, and does it align with the known history of the introduction? So in order to do that, we did a demographic reconstruction using this program called GONE, which is used to estimate recent effective population size changes. And we'll be comparing the Hudson and the Columbia River. So Hudson is the ancestral Atlantic, Columbia is the introduced Pacific population. And when we do that, we actually see a pretty clear signal that our Columbia River population has undergone a major bottleneck in the recent past before recovering a little bit more recently, especially relative to the Hudson River population. And when we include the actual date that these fish arrived in the river, it corresponds quite nicely with the effectively the bottoming out of NE in this system. If you look a little bit closer at just the Columbia River population and also include the census population size based on historical records, we see a cool pattern where the increase in census population, which is the light purple, also appears to correspond with a recovery of effective population size in the recent past. And I included this line that indicates the construction of the Dalles Dam because this is believed to have been facilitative in this population expansion. So what happened in this river system is that migration was effectively halted by this natural barrier called Celilo Falls. It was also a major indigenous fishing ground. And in 1957, there was the construction of the Dalles Dam, which not only flooded this waterfall and allowed access to a much larger section of the river, but then also enabled passage past the dam because these fish apparently really like to use salmon ladders. So that's a cool result, and it's really cool to see genetic data line up with the known history of a population. So do we actually see patterns of genetic structure? So in order to get at this, we can do an admixture analysis, looking at clustering across all the sampled individuals. When we do that, we see a pretty clear signal that these specific populations are genetically distinct from their ancestral counterparts. So depending on the value of K you're using, we can separate out both the Pacific and the Atlantic populations, and then can further segregate our Pacific populations into this northern Pacific cluster, which is in green, and a southern Pacific cluster, which is in orange. And we found K equals 3 to be the best supported amongst the different values of K that we tested. So if we use a PCA of individuals across using whole genome data, we see a similar pattern where we see 3 distinct clusters, and we see the southern Pacific kind of clustering amongst themselves in orange, northern Pacific in green, and then our ancestral Atlantic all clustering together in the blue. And if we color by individual rivers, we actually do see a signal of fine scale population structure, at least a little bit, which indicates that these fish are returning in general to the rivers that they were born in, at least in the southern Pacific part of the range. So finally, do we see evidence for local adaptation? So we're looking for these islands of divergence between ancestral and introduced populations. So in order to get at this, we can actually use our admixture analysis as a baseline expectation for allele frequencies across the genome. And what we're looking for is sharp allele frequency differences between ancestry components. So in this case, we're using the tree for K equals 3 that Ohana had generated, and then scanning for selection on each of the branches that leads to the northern and southern Pacific clusters. And when we do that, we do actually see several outlier regions pop out in both of these groups. So if we look more closely at just the southern Pacific cluster, we find a number of genes associated with these outlier regions. The bottom plot is ZFST, so this is just FST between one of the South Pacific populations and the ancestral Atlantic. And we can do the same, and when we do that, we find overlap with 139 genes. And if you can do the same with the North Pacific, you see a kind of similar pattern where you actually see some parallel signals in terms of similar regions appear to pop out between the North and the South, and you also find quite a few genes overlapping with these outlier regions. So we see 236 genes in total. So I'm sure you might have noticed this already, but there's this big block of differentiation on chromosome 6 that pops out in both of these plots. And this is what we believe to be a fairly large inversion, which is about 20 megabases in size. And the fact that it's even popping up in these FST plots indicates it may be segregating spatially. So what we can do is we can actually look at the different inversion haplotypes across all the rivers that we sampled. And there's a lot to kind of digest here, but basically you see different frequencies of haplotypes in different rivers. And if we focus exclusively on the snake and the klamath, we see that the majority of individuals are homozygous for the inverted state. And the interesting thing about the Snake River in particular is that it's not actually an independent river system from the Columbia. So the Snake River is actually a tributary of the Columbia. And what we're looking at actually is sampling about 700 river kilometers further upriver in the same river migratory complex. So the Columbia population was sampled at Bonneville Dam, which is closer to the mouth of the river, and we see all 3 inversion haplotypes present. But if you sample at the Snake River, which is, again, 700 kilometers further upriver, we see basically they're almost entirely either heterozygous or homozygous for the inverted state. So our current working hypothesis is that we're seeing a filtering effect of inversion haplotypes by migration distance. So this inversion might actually be important for migratory ability over long distances. So we're not entirely sure yet. We'll have to do a little bit more work to actually better characterize this inversion, but that's kind of the idea that we have for now. So in summary, we find that invasive shad are genetically distinct and also less diverse than their ancestral populations. And selection scans indicate evidence for locally adaptive genomic regions, including a big chromosomal inversion that appears to have been inherited from its ancestral population, but has since been segregating between rivers. And overall, despite a significant loss of diversity, invasive populations still show genomic signatures of rapid adaptation to new environments, indicating that genetic diversity isn't the end-all, be-all. As long as you have diversity in loci of high effect, you may still be able to evolve quite rapidly in a new environment. So with that, I'd like to thank my advisor, Nina, Dan, who collected many of the samples as part of his postdoc, and then allowed us to do whole genome work with down the line. And then, of course, all of the various collaborators, including the Karuk tribe of California and the Columbia River Intertribal Fish Commission, for allowing me to use the samples that they collected for me. So, thanks. Yeah, so like there's kind of two different approaches. One is kind of more functional, where looking more closely at the genes that are actually located within the inversion and see if there's any sort of obvious candidates that are associated with what you'd expect in terms of, maybe metabolic activity or muscle cell fate. The other thing is actually just looking at different forks of the Columbia and further upriver and see if we actually still see that inversion at long distances, and kind of see if we see the replicated signal of longer migration distances and inversion frequency. So kind of two different ways to look at it. Yeah. Yeah.
The migratory evolution is really interesting. Can we get pictures of those fish and do like a morphometrics analysis?
Yeah, that's another great idea. Yeah. So with the samples that we have here, we just have fin clips, but I am planning to do some additional sampling at the dam where they're found, and hopefully you can just get whole fish from them. Yeah. Oh yeah, I'll just do one more question. Yeah.
Okay. Great talk. Did you look at the inversion frequency on the East Coast?
Yeah. So the inversion frequency is actually on this plot here. So if you look at the blue populations with the bar under, those are all on the East Coast. So actually you do see some segregation between rivers that are relatively close together. So not entirely sure what's going on there, but it's a cool pattern. Yeah. All right, cool. Thank you.
Alrighty. Kia ora. I'm Kat, and thank you for being here in the second last talk of the entire conference. Drinks are so close. So I'm a postdoc working in Australia and New Zealand, and I study rapid adaptation, invasive species, and the focus of my postdoc has been primarily on non-SNP genetic variants. So in invasion population genomics, genetic diversity is our bread and butter. So we use it for a whole bunch of different things, like quantifying the impacts of genetic bottlenecks, translocation events. We can also look at changes in phenotype and whether they have underlying particular genetic variants that may explain them. And we can look at genetic diversity patterns across the landscape to determine how abiotic factors may be changing invasion speeds or range edges. But ultimately, all these studies rely on, to some level, a quantification of genetic diversity. And by studying these patterns, what we're hoping to do is understand more about the adaptive potential of invasive species, understand why some populations are persisting, with the idea that we can bring what we find both to manage the invasive species, to learn more about fundamental science, and maybe also we can apply some of it to those other species that are not doing so well. But what exactly is genetic diversity? Well, genetic diversity is kind of what we make it. It kind of depends on how much money we have, what tools we have to quantify how the genomes of one individual in a population are different from the individual genome of another. So at the moment, the primary method that we do so is through SNPs, which are usually applied in a reduced representation setting, though sometimes we quantify them in a whole genome setting. And these are the absolute smallest types of genetic variants, the absolute smallest ways in which a genome of one individual in a population can be different from the genome of another. Now, if we consider this on a huge spectrum of all the different types of genetic changes that you can see, this is a tiny little sliver. On the other end, we've got the big changes, the chromosome and entire genome duplication events. But luckily, those are mostly plant people's problems, and we don't have to worry about that. The subtext here is I'm starting a postdoc in about a month's time to work on plant genomics, so I will be suffering with you very soon. But as we've heard at this conference throughout the last couple of days, structural variants are probably the coolest thing since sliced bread, because they take up a vast majority of this spectrum. Structural variants are huge and they are important. They are traditionally defined as somewhere between 50 base pairs long up to potentially millions of base pairs long. And these are differences between individuals that are deletions of entire sections, duplications, inversions, things going to a different chromosome somewhere. And they're very complicated to study because it's taken us till now, till the time that we can afford to do whole genome resequencing to fairly high levels in order to study them in a broad genome-wide up way. Previously, we've been able to study things like this, but often it's because we notice there's a giant pattern, usually a large inversion, and those are traditionally the types of structural variants we've successfully been able to study. Because their size means that they often span more than the actual length of the reads that we're using to quantify them. So, ideally, what you need is whole genome resequencing to quite high depths, though that's sort of the baseline. Above that, you've got long-read sequencing, and the absolute gold standard, of course, is the 100 different telomere-to-telomere genomes that we have, all for the same species that we all have lying around our back pockets and can afford. So, structural variants are very difficult to quantify, but they are important. Their size means that the likelihood that they are neutral is much less than SNPs. So we want to quantify them. To what extent is it important, though? Can we not simply infer the genotype or the existence of a structural variant from its surrounding SNPs? And the answer for that, very unfortunately, is no. Both for biological reasons, they often occur in regions of high recombination, and also for technical reasons, because the existence of a structural variant sometimes stuffs up the SNP calling around it. And also, think about the PopGen pipelines you've done and the SNP filtering you've done. What is one of the most traditional SNP filtering steps that we do? We filter for read depth, not just minimum, but we filter out the maximum. Anything above a couple of standard deviations gets booted out. And you know what have a lot of repetitive regions with high depth coverage? Structural variants. So we're naturally excluding these from our data sets. So structural variants are very important. We need to study them. And inside structural variants contains a great amount of variability in what a structural variant is, as I've touched on: size, the type of variant difference. And I would say there is another really big important subclass of structural variants, and those are transposable elements. Now, transposable elements are same, but different. SVs, they arise in the genome because essentially, I hate to break it to you all, but you're all imperfect beings. Your mechanisms around DNA replication, repair, recombination, they're not right. They're mistakes, and that's why the structural variants arise. Ts, on the other hand, are traditionally called jumping genes or genome parasites, and they self-replicate within the genome, or they hop around in the genome. And so they are important sources of genetic variation. But if you thought structural variants were difficult to quantify, transposable elements are even worse. Within all of you right now, the most common T in the human genome occurs 1 million times. How do you map that using read data? And how do you know if it's occurred 1,000,001, or whether you've got a new variant somewhere in you 1,000,001 times? So I want to I forgot to mention that Ts, within the context of invasions, are thought to be particularly important. SVs may allow the organism to have a bigger range of sort of functional genetic variants because they're less likely to be neutral. Ts, on the other hand, often replicate at a much higher rate than SNPs and SVs. And also, they're implicated in sort of short-term changes that may allow invasive species to, after they come to a new area, plastically respond to the environment. And also, stress can cause Ts to replicate within the genome, possibly creating more genetic novelty after they're introduced to a new range. And so the next time that you're standing up on stage, a little bit stressed giving a talk, or you've left writing your grant to the last moment, stop, take a breath, because every time you are more stressed, you're letting the Ts win. They're propagating in your genome, and soon they will take over. So Ts, SVs, they are similar. We need to consider them together, but they are a little bit different. We can have Ts that are non-variant within a population. We can have SVs that have nothing to do with Ts at all. And then we can have the other messy things. SVs that actually the entire SV itself is a T, or you could have a T embedded in a big SV, Or you could have a T that was like slightly upstream of SV. It's a little bit messy, and sometimes it may be appropriate to sort of parse those out and look at those biological processes related to invasions differently across these different types of genetic markers. And so, the questions from this then come, is studying SVs worth it? Do they tell different stories from the SNPs? And then if we decide the answer to that is yes, is it then worth splitting out SVs and TEs as two sort of different types or genres of genetic variants? And I'm going to tackle the story using two different species. Only one of this is an invasive. See if you can tell from its distribution map which one it is. So this is the New Zealand stitchbird, the hihi, and it currently exists naturally on one remnant population, which is the source of many mainland reintroductions. And now it's not an invasive species, but it's a small population, which, let's face it, is the other side of the coin to invasive species. So these are the ones that went through a bottleneck. They just weren't as successful. They wish they were us. But the cool thing about conservation and people studying these species is often they actually care about keeping them alive. And when they find them in the field, they don't immediately shoot them or pull them up from the ground, which means we have lots of really cool data for them. In this case, we have lifetime fitness, the total amount of individuals that were produced from one individual that grew up to become adults. So we have 30 individuals, we have short read data, we can quantify the SNPs, the SVs, and we can then relate that to lifetime fitness, specifically by tackling questions around load. Genetic load is the accumulation of putatively deleterious alleles within a population and is something that is really of concern in small populations and invasive populations after they go through a genetic bottleneck. We quantify the masked load, which is the heterozygous load, and the realized load, which is when the putatively deleterious alleles in the homozygous state, across SNPs and SVs. And we find, as to be expected, it's not completely that they don't agree with one another, but there is variability. It's genetics, there's noise, it's biology. We had to expect that. But what that means is we can't predict the structural variant load from the SNP load. And in these inter-individual genetic differences, the lack of predictability there, it means that there are real-world biological consequences from that. If we look at each of these 4 different load measures, SVs and SNPs, masked and realized, against lifetime fitness as individual models, they all have significance and they all agree in terms of the patterns. As, for instance, realized load increases, the overall lifetime fitness decreases, and then vice versa for masked load. However, when we analyze these in joint models with realized and then masked load, what we find, and another thing to mention and call out here is that you can kind of see a little bit of a spoiler, that the slopes for the SV loads are a little bit more steep and fun, which means that in those joint models, actually, the only one that came out as being super, super important or still significant was the masked SV load. Now, that doesn't mean the other types of loads aren't important, but what this does tell us is that the structural variants capture important and unique components of the genetic variants that we should be paying attention to, especially if we're tackling a population where we're doing breeding regimes or translocations, because these SVs, there's only 400 SVs, there's millions of SNPs. These are the ones we really should be paying attention to. They're probably very high impact. So, do SNPs and SVs tell the same story? Yeah, nah, unfortunately, as we say in Australia. What about SNPs, SVs, and Ts? Well, I should say for the first study, we deliberately excluded any SVs that had T elements to it. We just wanted to exclude it for the purpose of that study. With this next study, we used an invasive species and we quantified the SNP, the SVs, and the Ts independently, making sure that none of those overlapped. And so for this study, we used the invasive minor, which is an excellent study system because there's a whole bunch of sequential and sometimes parallel introductions across the globe. Somebody thought that was a good idea back in the late 1800s. Conservation geneticists look at that and they go, who the hell thought that was a good idea? And invasion geneticists look at that and be like, who thought that was a good idea? Anyway, it's a bit unfortunate, but it is a natural system, or a semi-natural system, and a really cool experiment that we can ask these questions over. How do these 3 different types of variants be impacted by the bottlenecks in this? So we see across this that there's a slow accumulation of the sort of runzo homozygosity, the inbreeding, as the sequential bottlenecks stack up. And we can look at the structural variants in the Ts across these different lineages. And this is just a sort of a site frequency spectrum of them all stuck together. And the one thing that really jumps out to us is that the structural variants have a distribution of alleles, of rare alleles, that matches more the grey SNPs. Whereas Ts, we see that bimodal distribution. We've got the sort of variant ones on the left, And the one on the right, the higher peak, those are probably the Ts that are intrinsically part of the species with a little bit of dropout from the fact that it's short read data and we weren't able to detect T, maybe a specific T across all individuals. But if we look at these patterns across those sequential bottlenecks, what we see is that occasionally, when we look at patterns, the SVs and the Ts look like each other. This is a quantification simply of how many variants there are on average within a population. SNP's different, SVs, Ts look kind of similar. Quantifying this pattern in a slightly different way, looking at it again in a site frequency spectrum-like analysis, focusing on the rare alleles, what we see is that it's the SVs and the SNPs that look more similar and Ts look a little bit out on their own. So clearly there's an interaction between the types of genetic variants that we're dealing with, their properties, how often they arise in the genome, and the demographic processes. And so the answer to the second question is also no. So to quickly finish off, I guess I wanted to address firstly the invasion biologists in the room. If you're sitting on whole genome resequencing data and you're thinking about, should I analyze SVs? I say definitely yes, but be prepared for a lot of manual curation, especially if you're using short read data. But analyze it because it may give you extra information about why exactly your species was successful and how it's adapting in the new environment. And vice versa, by studying these processes in invasive species, we're going to be learning more about how these different genetic variants respond to demographic and evolutionary processes. And finally, to the SV people in the room, my question to you is, and this is a very open-ended question, how much do we care about false positives and negatives? How much do you think we should make identifying SVs and studying them accessible to more people so that people can reuse their whole genome resequencing data in a way to tackle questions in a big way across many taxa so that we can start getting to some broad truths? Because it's early days in SV analysis, and I think if we're too restrictive and we say we've got to wait for whole genomes or population level long read data to study these things, we're going to be waiting a long time to study it in anything but humans, Drosophila, and maybe counts. Anyway, thank you all for listening. I probably have time for one very quick question, but please grab me after if you have any more.